Good afternoon, everyone. It looks like a few of you are coming in from the waiting room now to this Water at Wayne session. Thanks for joining us. We will be getting started in a few minutes. We normally like to wait 3 or 4 minutes to make sure everyone has a chance to get in before we kick it off. So again, we'll be getting started in just another couple minutes here. All right, folks. I think we are about ready to get started here. And so let me just say once again, thanks to all of you for joining us for this Water at Wayne session. We will be hearing from Judy Westrick in just a few moments. Before we get started, let me give you some of those housekeeping tips that I think everybody knows now regarding Zoom. It's going to be best if you keep yourself muted throughout the event today, unless we have a little extra time towards the end and Jamie opens it up to questions. But otherwise, please keep yourself muted, and I think we will do that somewhat automatically for you. We'd ask you to keep your videos off throughout the event so that we can spotlight Judy and her slides. We're hoping you have questions, and as questions come up, please feel free to just toss those into the chat box. We'll be monitoring that chat box, and if it's something critical that we need to get asked right at that instant, we will go ahead and ask it at that point. Otherwise, we may just keep the questions until the end. So again, thank you very much for joining Water at Wayne today. I'm very much looking forward to this presentation to hear some things that I may not have known before about the Lumigen Instrument Center. I know that they've been really ramping up over the last year or two to expand their environmental research capabilities. And that's been a real plus for Healthy Urban Waters, as well as the CLEAR group, the CURES group, and the rest of our environmental programs here at Wayne State. So without further ado, I do want to introduce Judy, even though I know many of you are very familiar with her. She is the director of the Lumigen Instrument Center here at Wayne State. She's an accomplished environmental analytical chemist whose research focus is on cyanobacteria and their toxins. She has conducted cyanotoxin research in a variety of areas, including determining the occurrence and drinking water treatment, removal, and/or inactivation efficiency of cyanotoxins, interpreting policy, and identifying knowledge gaps between academia and industry, as well as developing standard analytical methods for the priority cyanotoxins. She has organized and served on local, national, and international cyanotoxin panels. Her current research includes developing simple, robust liquid chromatography mass spectrometry methods to quantify cyanotoxins and persistent organic pollutants in water and tissue. Her background as an analytical chemist, a certified drinking water operator from 1995 to 2001, and a cyanotoxin expert, has enabled her to be a linker between the academic, industry, and environmental health communities. So happy to have Judy here talking to us today. And Judy, I hand it over to you.
Well, thank you very much, Carol, for the nice introduction. I guess I'm going to start with, can everybody see my screen at this stage? Carol, can you, we're good. All right. That's all I wanted to make sure. So Judy Westrick again. I am the director of Lumigen Instrument Center. And I'd like to start off with kind of a basic introduction of the Lumigen Instrument Center, which we commonly refer to as LIC. The LIC has three ways in which people are currently using the LIC. So the first way is what we call user-based usage, which is when somebody comes up and says, we need to use your LC mass spec. And I would like to do it. I'm a graduate student. I'd like to get the expertise and run my samples. So then we basically go in the process of training that person and then getting them to run the instrument on their own so that they have it to basically do their analysis, whether they want to do midday or in the middle of the night. They also have that capability now putting on their CV that they can run an LC mass spec or whatever instrument they're running. So that's what we call user-based. The other way is what we call fee for analysis. And that would be when somebody drops off a sample and says, we'd like you to run this method. That we'll pull up the method, we run it, and then we just send the results back. We have our QA, QC, we have all those discussions, we have a meeting, but it's basically what we call fee for service. The third way is the way I'm going to talk about a little bit today, or the three projects, is that I call it kind of where we actually either bring expertise into the field. So you're running a project and you need a little more expertise, let's say in the area of mass spec, and it's going to be a major part of your project, or electron microscopy, or whatever major instrument we have here. You don't have the extra staff that you need to actually troubleshoot the project or learn that instrument, or you're just short on staff, or and you need someone to help with that project. So the three projects I'm going to talk about today fall into that last category, where we have been incorporated into the research as a part of the team or a collaborator. And that way, basically, my scientists are working on that team, actively attending meetings and synthesizing ideas and putting them forward. So the three examples I'm giving today is the first one is on harmful algal blooms. You kind of heard in my little bio sketch that I like harmful algal blooms. The second one is the topic is going to be on VOC or vapor intrusion. And that's based through a CLEAR project. And the last one is going to be based on a healthy urban water / One Health project, which is going to be based on historical PFAS. You know, when did certain PFASs come in and come out? And this is going to be based from a sediment core. We're going to be looking at historical PFAS. So on that note, I'd like to start on my first project that the LIC has worked on. And we're going to see if we can advance forward here. Okay, so we're referring to this project as the HAB grab. And that means it's a harmful algal bloom. And it happens, the project location is going to be the western basin of Lake Erie. Harmful algal blooms are of concern because of both environmental and human health. The harmful algal blooms, the cyanobacteria, produce secondary metabolites such as microcystin, anatoxin A, slendrospermopsin, saxitoxin, and BMMA. Microcystin is a hepatotoxin, anatoxin A is a neurotoxin, slendrospermopsin we refer to as a cytotoxin, mostly focusing on the kidneys and the liver, saxitoxin, a neurotoxin, and then finally BMMA, which we call the new guy to the block, which we're referring to as a neurotoxin. Interesting enough is BMMA will incorporate into human, fish, whoever ingests the BMMA into their protein and replace serine. So hence it changes the folding of protein so it can have a large effect on many functions throughout the body. Mostly we see since it crosses the blood-brain barrier and the brain. Why are we interested in this? Specifically, Lake Erie, microcystin has been the topic. But what we really are interested in Lake Erie is to be able to give people a forecast, to give them an indication when those blooms are going to occur. In order to do that, in the past, we've monitored, we've supplied lots of data analysis, and we've tried to develop a predictive model. Our current model mostly focuses on the fact that we can just predict the bloom, but it's not the bloom that concerns us, it's the toxicity. So the idea now is to transfer the idea of we can predict a bloom is, can we predict the toxicity of the bloom and where the hot points will be? So on the right-hand side, you actually see some of the parameters that have gone into the past model, your nutrient load, the water temperature, how the water's flowing, how rough it is, how calm it is, whether you have thermal stratification, and the rainfall. The rainfall is the bigger one from the standpoint is that the water will actually bring in the nutrients. And we find that the current model that we use, the nutrient loads from the fall are more important than the nutrient loads from the spring are. All of these, when they are at the proper levels, end up with the harmful algal bloom. And you can see here on the right-hand side, this picture, this plum here. And this is actually microcystis. It produces microcystin and several of the other toxins. But in this case, this is a Lake Erie bloom. So this is actually one of the bays in Lake Erie. And you can see how pretty that is. It's so lovely. So the HAB grab happened in 2018 and 2019. And what it was basically, you'll see on the right-hand side here, a picture of the Western Basin of Lake Erie. And then over the top of that, you'll see dots. The dots are going to be what we call HAB grab, meaning we went out and grabbed samples all within one morning. And then the other area here is superimposed with the cyanobacteria bloom at the time. So the bloom we can see. The dots are going to give us the toxins and other parameters that we could potentially use to help us model. So the question becomes is, where are the toxins and how much toxins are there? This happened only once a year, 2 years in a row. One high year bloom, which was 2019, and a low year bloom, 2018. Dustin Chafin was the PI. He's from Ohio State. And this is actually a NOAA EcoHab funded project. The LIC group that participated in this mostly was Westrick, myself, Burbank, and Fur. Other universities you can see as they're all over the map here included University of Toledo, Bowling Green, Wayne State, a private firm called Linbo Tech that does research, University of Michigan, and Michigan Tech. And then for the 2019 study, Canada actually was allowed to come on board, hence allowing us to sample in the Canadian water. So you notice these are more, the Canadian waters are more separated, are further apart, whereas if you look at the water in the area down here, they're closer together. So what are our challenges when we look at forecasting going forward? It's like right now we have points that you can see these points in Lake Erie. They're far and few. And that far and few are where we take regular samples. So we will take samples either every day at some of these points or twice a week. We have no idea what's going on in between these points or how to interpolate in between them. Timing is important. So we wanted to make sure that we grabbed it during a bloom, and we got lucky. We got a high toxic bloom and a low toxic bloom. So we got lucky on that to help us try to model it better. The grid, the grid itself, which is both 2018, 2019, much of it started with how many boats can we get out from which universities and how close or far can we be to be within everybody's budget. Only part of this budget actually came from NOAA. Others donated their time. Other universities donated their time. Limnotech donated their services, which is huge. So that's how this all came about. And once, like I said, after 18 was done, 19 thought it would be a great idea. We had to create the SOPs for the sampling. Sampling that many sites, 200 each year is huge. Handling, analysis, and we're going to talk about those a little bit more. And then how to distribute even the equipment, which all happened relatively smoothly. So, well, here's our pre-labeled bottles. Everybody's on the boat. They're handed their bottles. They go out. They go to the GPS coordinates. They don't stop. They literally just shut the motor off the boat, but they don't anchor. They drop their stick. They pull up a sample. They write it on their sheet. They put it into 2 bottles, and then they take as many of the parameters that you can off of a YSI. And so you usually can get turbidity, pH, phycocyanin, chlorophyll, DO, and the last one would be particulate count, not count, but conductivity. So, and then basically they're on to the next site. This actually was the relatively easy thing once we got going. This was the hard part that nobody anticipated, was 200 samples arriving at University of Toledo at one time. So you have the 200 samples come in, and of those 200 samples, which are 2 big liters, so the liters get separated, you approximately have 8 labs they have to go to for different analysis. And then they have to be, some of those labs actually need subunits of that. So what you see here then is the bottleneck. And this basically happened. So in the morning, sampling was done. By 2 o'clock, it was over with. By the time all the samples came in, it was 4, and they didn't actually shut down either time they did this in 2018 or 2019 until 2 or 3 a.m. in the morning, until all the samples got separated. So this was the bottleneck and something to think about in the future. However, I'm going to state we all wanted all the samples to be in one location to undergo the same thing. If you didn't notice in the corner, I have where the funding is coming from, because I'm trying to make sure that everybody understands we're a multi-funded core. So NOAA was funding us to do that part. However, the instrument that was ran or used to run on this was funded by Cures. And that was a P30 grant that Wayne State has received. So cyanopeptide analysis was ran on our Altus, which is one of the most modern triple quad mass specs. We have online concentration. This allowed us basically to do those 200 samples very quickly. And John, so what do I mean by very quickly? We probably ran those 200 samples under 3 days and only running the instrument overnight. The good news is again, we use a mill in the online concentrator, and it goes down to all of the full mill goes on to the column. So that's our concentration, and we can go from there. So there's no solid phase extraction. The types of molecules we're going to be looking for is microcysts up here, which is that hepatotoxin. And the other primary one is going to be anabinopectins, which one is called acrylamide Y because it was actually found in oscillatoria, where the other anabinopectins were found in anabina as a genera. So sometimes, even though they're the same family, they have different names up front. So this is just to give you an indication of how well our analysis works. On the right-hand side, you have the 12 available microcystins. And then over here, we have our anabinopectin B, F, A down here. And then we have some of the other cyanopeptides that we ran. However, the anabinopectins and the microcystins were the ones that were most frequently seen. And I just wanted to show you over here that we get nice linearity for all of these. And it works well. And unlike the EPA method, we've gone a step further and used the European method, where we use what we call a quant and a qual ion. And that ratio between those, when we see them, has to be a certain number, or we start thinking that potentially, if that ratio isn't what it needs to be, that we actually might have an isomer of that microcystin. So what do we see when we actually put them back onto the map? Well, microcystins wise, of the 12 that we monitored, we see 4 major ones. And those 4 major ones are considered the major microcystins that are in the area, or in this area being the Midwest and the East Coast. That would be LR, RR, and YR, and more recently LA. And when I say more recently LA, if you would have talked to the people monitoring, like myself, the Great Lakes 10 to 15 years ago, and you said you saw LA in Lake Erie, they would tell you that that would be highly unlikely. However, if you told me you saw LA on the West Coast in California and you didn't see any LR, which is considered the prevalent one around the world, I would say that would be very likely. Why these are regional, we don't know. And we don't know why now, if it's climate change or if there's other factors to why now we're seeing LA, but LA is a prominent, has become a prevalent microcyst. Untargeted ones means that we actually saw these with no standards, and we actually really need standards for them. We have Alex working in our lab, our undergrad, working on isolating FR from one of the blooms, that actually the bloom you saw earlier. We would like to also isolate homophenylalanine microcystin. the other points that I want to point out is you'll notice that the microcystin is not evenly distributed through the bloom. You'll see that the microcystin basically concentrations are higher along the edge, but you'll also see the prevalent one is RR from time to time, and it actually it looks like it's RR all the time on this one. And if you would have caught it a little earlier, it might have been LR. The other thing is like I said, as you go out, you'll notice the microcystin dissipates. If we look at the aminopeptins, now this isn't really considered a toxin at this stage. However, we know it's antimicrobial, antifungal, and we know it also inhibits, some of them inhibit protein phosphatase like microcystin does. And it also inhibits carboxylpeptidases, which are digestive proteins. So as we look at these, again, we can see that our prevalent one is microcystin YR, which you can see is the big red bloom here, the big red line here. And what you see here is microcystin, aminopeptin B is the secondary. Also, you'll notice a difference in the distribution now. The difference in the distribution is that the aminopeptins, concentration-wise, are similar throughout the whole bloom. So whereas the microcystins were along the coast, here we have them throughout the whole bloom, which is telling us that the microcystis in this bloom is responding differently to its environmental conditions. So what do we know now from the HAB graph? Well, we can spatially interpret in between places. We can do that for aminopeptin and microcystin groups. We can, we've made heat bars or heat maps and bar graphs to show where the microcystin and the hot spots are. Microcystin concentrations are about 5 times more down here by the shore, whereas as you go out, they're about the same concentration out here, which is about 2 parts per billion. And we actually have identified several new microcystins, and that paper is going to be published here shortly with Dragon from the University of Toledo. So that's the end of the talk on HABs. Now we're going to move to vapor intrusion. And this is sponsored by CLEAR, which is a group on campus that does integrative research. And you'll see again the CLEAR symbol over here in the corner. The primary research PI on this is Tim Dietrich. When he was approached to basically work on the mitigation for vapor intrusion, he decided he wanted to develop absorbents and VOC absorbents specifically. And he was at that time short-staffed, and my staff was very capable of doing this. I worked in the area of VOCs and media and carbon, granule activated carbon, when I was in the drinking water industry doing research on that area. Nick from the lab, Nick Pereno, he actually did most of the work. By trade, he's a chemical engineer, so he had no problems understanding absorption and the process. And he also is a mass spec expert now after being here 3 years. And he also has a PhD in biomedical research. Our collaborator on this was Yang Li Wang, Wager. And then we have students, undergrad and graduate students. So it's a big group and a big team that we're working with. So what is vapor intrusion over here? So basically what we have is contaminated soil. And that contaminated soil is basically going to start releasing VOCs as vapors, and those vapors then can be found exterior. And if you have cracks in your house, in your basement, the VOCs then can be released into your basement. If it's being released exterior, there's actually areas in Kentucky that you can monitor vinyl chloride, which is a VOC, outside on top of the land where the vinyl chloride was buried. In Michigan, what we're finding is that many of the VOCs are coming in through the basements and cracks and then being concentrated because the air isn't being turned over in the house. So why VOCs? Why BTEX? Why TCE? Because in 2018, there was a big write-up in Detroit News, which just kind of highlights why we wanted to focus on this. And the reason we wanted is that basically, thousands of Michigan sites need to be tested. 276 Michiganders were basically ushered or barred from using their homes or work sites. That's a lot of displacement. And the impact was mostly in Detroit, Grand Rapids, and Sturgis, Michigan. Detroit, of course, of interest of ours. One of the major ones in 2016 was a preschool was closed where 224 people were moved from the site and 80 of them were children. And Eagle estimates that over 4,200 possible vapor intrusion sites are still out there and significantly influencing public health negatively. So what was our part of the project? There are several parts of this project. There are biomedical parts of this project that are actually looking at in pregnancy and exposure to VOCs. And there's parts with in the area of looking at genetic changes, again, in fetuses. And then there's areas also looking at monitoring groundwater. However, ours is going to focus on the absorptivity of medias. So there's 4 medias we're going to look at. We're going to look at Sol-Gel, bone char, activated carbon, and zeolite. So the first job is that Sol-Gel is an area of expertise of Tim Dietrich, and he basically wanted to be able to synthesize the Sol-Gel and say that yes, we can synthesize it, and we can synthesize it similar to the commercial available products, and then we're going to alter it to do better than the commercial available products. In order to do that, we had to draw a baseline. So we basically looked at the SEM of the absorbent materials, and then we looked at the absorption kinetics, and we looked at isoforms. So basically the synthetic process here was we started off here with like a monomer material. This is methyl cyanol, and we basically catalytically reacted it to make a gel. And then we capped the OHs at the end of the gel. The capping is important because if we don't cap it, then we don't get the right swelling characteristics. So once we stepped through the process, we actually used some things that we had left over from the person that I took the lab from. So we basically cured it for a week when we put everything in together. And then it says we cleaned it by Saxolite. So the Saxolite actually was left here by one of the analytical chemists I took the lab over from. And then we basically recovered our Sol-Gel material, which is really light here, but you can see it sprinkled. And so basically 95% of it was above 250 micron pieces, which then we could grind and make and sieve to our desired sizes. So what does it look like? This was produced by Dr. Mike and the EM lab. And so, oh, by the way, the synthesis was done by Nick. His PhD was in synthetic chemistry. So we looked at it under the microscope, under the SEM, and we see activated carbon. We see the tubes, the carbon tubes, and we could validate it by EDS that it is carbon and mostly carbon. We looked at the zeolite, and we could see the structure of the zeolite, which is more like packing, like slats of packing. And then we looked at it from the EDS perspective. We saw 60% oxygen, 60% silicon, and 8% aluminum, which is what we expected in zeolite. And then this is the commercially available Sol-Gel, which is called Ozorb. And we could see basically it looked like, I call it little cauliflower, and with micrometer size pores, so lots of pores, lots of open space to bind to. And basically the ratio of oxygen to silica was 2 to 1, and we know these vary, so it was about 2 to 1. So then we produced the our Sol-Gel, and you see it here. It looks very similar to the Ozorb. And then here we have Sol-Gel with a T-hexyl capped. So T-hexyl caps that oxygen. These spheres have these guys have finer nanostructure fuzziness. So it looks like we actually have better nanostructure here. So before I get going to how we actually did the actual analysis themselves, I will talk a little about the instrumentation since we do this. Nick was responsible not only to do the synthesis, but to run some of the analysis and develop the analytical tools we needed to do the quantification. Nick used the GC mass spec, and then the beauty of having Nick was that we went through two auto samplers, because the first one I think is 20 years old and the second one's 25 years old. So I ended up having to borrow one. They're obsolete. So Nick went in and made them work, which was nice. And this is our purge and trap apparatus. So the purge and trap is connected to the GC mass spec. So we use what we call dynamic headspace, and I'm going to put you over here. So what you have here is an example. On the top, you have air. On the bottom, you have water where your VOC would be. The needle that comes into here has a hole down here where it basically allows you to purge through the water. And then up here, it basically the VOC comes in and then it pushes through here the needle and comes back out here into the valve and then into what we call a trap. This trap is a K VOCarb 3000, and then it sits in, and it collects in the trap. So that allows us to take a very dilute sample down here and concentrate it here. Then when you come down below, now what you do is you say, I want the sample. So it's VOCs, you heat it up. When you heat it up, here, it goes into what we call the dew stop. So it's a little cooler there. It drops out the moisture and then it goes into the sample.
Hey, Judy.
Yes?
Your mic is going in and out a little bit.
Oh, I'm sorry. That's what I was worried about. Thank you for telling me. So basically it comes out into the GC.
You're good.
All right. So calibration curves, again, we just wanted to show that we run our calibration curves. We have them for the various VOCs, which is going to be BTEX, benzene, toluene, ethylene, benzene, and xylene. And then we have over here, we just show you that our low concentrations, and high concentration, what type of error you might expect to see on them. So this is what our standard curve was, but then this is what our sample would show. So 14% off. The next thing is then we did absorption kinetics. So before we do isotherms, we have to understand where equilibrium, how long does it take to get equilibrium? So what we see here is on the bottom, minutes, and on the side, basically how much was absorbed. And when we get to equilibrium, it'll be flat up here. So what you notice is the bone char over this whole time period, which is 24, about out in here, 24 hours, you still have absorption going on. So it's very slow. On activated carbon, you'll notice that at time 0, you're here, then at about 2 hours, you're still absorbing, but then after about 4 hours or so, you're flat. You're basically at equilibrium. For the Z-Lite, we have the same thing going on. It just keeps going over the time course, so somewhere around 20 hours. And then the commercial Sojel, notice that on the left-hand side over here, notice that the equilibrium time is only 2 hours. So that's actually really good from a loading perspective. It's a very quick loading. And then notice the Sojel that we made, it's only also 2 hours. So they're very similar in that respect. And also notice that each of these, so this is MP xylene, and then benzene, and then going down toluene, ethylbenzene, and TCE. Notice that our left axis of concentration is the same, and notice that absorption for those are the same. So that's another thing indicating that we did a good job making the same thing they had. The toluene isotherms, we only showed one set of isotherms. That one set of isotherms, if you look at them, when we look, we did them under different conditions because we know that the characteristics in which things absorb is going to change, could potentially change, or likely will change under different conditions. That also gives us a little information about potentially what capabilities or how things are absorbing. So toluene and DI water, you'll notice that actually the Ozorb works the best. So the steeper the line, the better. Let's go over the axis quickly. In how this experiment's ran, is you have a vial, and in that vial you put water, a known amount of absorbent, and then a known concentration of the toluene. And then you take another bottle and you put the same amount of absorbent in it, and you put a different concentration of toluene in it. And that's why you have down here toluene changing versus over here, you're going to have initial concentration versus final concentration divided by the mass of the absorbent. So in theory, your absorbent's always going to stay the same, and the changes are going to be from the initial to final and versus over here for the final. So the steeper, the more concentrated it is, and the shallower it is, the steeper, the more it absorbs, the better it absorbs, and the more shallow, the less it absorbs. And you can see that the OZORB works best here, then GAC, then ours. And then when we get into the brine, so the saline type water, you see OZORB, the Sol-Gel we made versus GAC. And one thing you'll notice here that the GAC and the OZORB and the one we made are very competitive, and they seem to stick the three together. And then we come down here at pH 13. You see that the OZORB and the GAC, and in this case, ours has decreased a little more than normal. And then over here, again, the GAC. So competitive, again, the one we made is competitive. It's from a isotherm perspective, but it isn't quite as good as the OZORB, so we're gonna have to work on that. And sometimes the cheapest form is GAC, and sometimes the GAC outcompetes both of them. So in summary, the reason that we're looking at this is Detroit area has brownfields with soil VOC contamination. That contamination can be industry, it can be a private business such as dry cleaners. So oftentimes TCE is seen where old dry cleaners used to be. Many of those are brownfields. We don't know where they are unless you go back and look at old maps. VOC intrusion is a real problem, especially in urban areas, in Detroit specifically. So what we've done as part of the Clear Environmental team is we, as a group, have synthesized the Sol-Gel absorbent. We validated a dynamic headspace so that we have analytical methods to move forward as we alter our Sol-Gel absorbent. And as we synthesized it and compared it to other products. And that's the end of our Clear project. So the final project that we're going to talk about is a historical perspective on PFAS contamination using sediment cores from 2 lakes in Michigan. This is sponsored by Healthy Urban Waters and One Health. So this is internal money to develop a bigger plan or some future, hopefully, grants and publications. So the 2 lakes that we're looking at in Michigan are very different. There is one up in I call the tip of the mid area in the middle, and that's Mayo Dam. Mayo Dam Pond is actually a dam pond along the Asable River up here, and it's pretty isolated. Down here in southeastern Michigan, we have Ford Lake, and Ford Lake is located in Ypsilanti, and it's along the path of the Huron River, which actually eventually empties into the Detroit River, or where the Detroit River is going into the western basin of Lake Erie. So this is also a big team. I would say the PI on this is Carol Miller, who introduced me this today. And the project leader is Jamie, who is the person that's moderating. And the other people involved on this, it's again, we have a core. And so we're looking at land use and history, Dr. Mohammed. Plastics are being looked at Dr. Wager and Cashin. The cyanotoxins, which I can tell you a little bit about them, but we're not done with those yet, are done by my lab, or LIC. PFAS is being done by LIC, which is the primary talk of this conversation. And resurrection ecology done by Donna Cashin, and magnetic susceptibility done by Dr. Seligman. So the core will have all of these analysis done on them. And then we will be able to compare them when we get all the data put together. So PFAS, where and human health question mark. Why is it such a big deal? Well, it's a big deal for several reasons, but let's just start to the left of my slide. Over here, you'll see to the left of the slide, you'll see that we have that's PFAS. That's home from PFAS, and that's actually one of our local lakes. So that's of concern. That PFAS would be in parts per 1,000 levels, so it'd be very high. You don't need a good mass spec to see that. One of the things that we're seeing out in the rural areas is high PFAS levels, like around Tawas or around Southbridge Field, is because there was a lot of firefighting practicing being done there. And one of the number one PFAS flame retardants for a while was PFAS. So we see a lot of areas that where like airplanes were, or areas like where firefighters would actually practice putting up fires, would have high PFAS levels, or potentially areas that actually had large fires before we knew PFAS was a problem. Besides firefighting down here, foam in the far right-hand side, where else do we see PFAS? Well, we see it in food packaging. We see it in outdoor gear. This is my favorite thing. I love my Gore-Tex coat. Hence, Gore-Tex is PFAS or some form of PFAS, some polyfluoroalkyl compound that allows it not to get you wet. Non-stick pans, stain resistance for your furniture, for your clothing, for rugs, and household items. It's also a good lubricant or surfactant. So where are we seeing the dangers in PFAS from a human exposure aspect? So increased risk of thyroid disease, high blood cholesterol levels, increased blood cholesterol levels. And here's the interesting one: decrease the body's response to vaccines. This was actually seen very early on, and I was in one of the Oceans and Human Health meetings when this was presented. It was, at first people kind of shook their head and said it can't be, because it just seemed like, how could a chemical make the vaccines not work? And what we were seeing up there is standard vaccines in the Inuit population, which had very high levels of PFAS in their blood. Basically, the vaccines weren't taking, and they were getting the illnesses that the vaccines were supposed to protect them from. We're seeing decreased fertility in women, risk of high blood pressure and preeclampsia, and then low infant rate. So we're very interested in keeping PFAS, even though it is persistent chemical that'll be around for a long time. We want to decrease that. And we want to know where it is. So Elliot Furr in the lab was the technician that was put on this project, and he developed a method to which we could extract the PFAS from the core. So we start with the core, and we remove about 200 milligrams, put it in the vial. We take the vial and we actually lyophilize it to remove all the water. Then if you notice, there's beads here. We extract it with a 70% acetonitrile and water. We spin it down. We remove the top. We blow down the acetonitrile and the acetonitrile and the water to dryness, and then we resuspend. We resuspend. And when we resuspend, we actually do it in 50% water and 50% methanol, because if you don't put the methanol in, the PFAS sticks to the glass and your concentration levels will change. And then we run it on the instrument that basically cures what for us. So this is, again, the Altis. The beauty of the Altis, you'll see in this slide, is here's our 16 standard PFASs. I think now we're up to 20 something. We add them as people want to use them. So we don't just randomly add them. So if somebody says, we'd like to add this PFAS, then we put it into the method and work with and get it in. These were the standards when they came out back several years ago for US EPA. You can see we get very nice separation, but this is the impressive part. I talked about online concentration. For this instrument, we don't need to online concentrate PFAS because it seems to do very well. So with no online concentration, a 50-50 dilution in water and methanol, we actually see PFAS with detection with minimum detection limits of low parts per trillion. This is parts per trillion, and minimum reporting levels at basically slightly higher than that. So this is phenomenal because this instrument has allowed us to remove a large constituent of the work that we would do in-house. So thank you again, Kira. So what did we see? I'm going to start with the cores. Were dated initially, and so here's the date timeline at the bottom. So we go from low 1890 to 210. So 1890 to 210. I don't want to lose my voice as I turn and look at the screen. I'm going to quickly review some of the things that you see up here. So basically in 1940, we considered the initial products production of PFAS. And by 1950, they PFAS resistant products were well on the market moving forward. In 1980, we had firefighting foams like hit the commercial market huge. And then in about 2000, you see US requesting industry to reduce the amount of PFASs out and present. So what type of PFASs do we see in our cores? Well, in 1890, like we're supposed to, we see nothing. Remember, cores are approximate. In 1910, we're starting to pick up PFAS. So this is the actinoic aspect of this. As we move to the 1930, we see what we call, I'm going to call ethyl FASA, which is right here, ethyl FASA, and we see PFAS. Now, I'm going to share with you, ethyl FASA does break down into PFAS, and then we see, again, here we see the ethyl FASA and PFAS, ethyl FASA, and then it stops, and then we just pick up PFAS. So the question is, I think what we're picking up here in Ford Lake is potentially some of the firefighting and some of the other things going on. And over here, we're not sure what we're seeing, and we're going to talk just a little bit about that in the next slide. But notice this is only Ford Lake, and we don't have anything for Mayo. Why don't we have anything for Mayo? Because Mayo had nothing. We saw everything was not only, it was below the detection limit, so we could not detect it at all. So we only have one. And this is the one that's in Ypsilanti, which is downstate. So where we're going to end up on this is, why do we see this ethyl FASA? Well, ethyl FASA is a precursor for PFAS in the actual process. It's also a ethyl FASA also will degrade to PFAS if it's in the right biological setting. We also know the ethyl FASI will oxidize into the FASA. So this is a part of the puzzle that we're going to have to study, and we're going to have to use land usage, and we're going to have to pull the other pieces in together to actually start deciphering why we're seeing this ethyl FASA. We don't actually monitor for this. So remember when I mentioned that we would be looking, potentially adding things, this is one we probably will add to our list of PFASs in the near future. So in summary, no PFAS was extracted from the Mayo Dam pond. So that's why you didn't see any data on it. 9 out of the 16 PFASs were detected in the sediment from Ford Lake. The 2 prevalent PFASs were PFAS and ethyl FASA. Ethyl FASA was started in 1930 to be detected and was detected to about 1970. And then it stopped. And then we saw PFAS with in 1990 to current. So I'm going to give you a quick overview on some of the analysis that we run for other people, just to give you an indication of the type of work we're doing now. And then I'm going to wrap it up. So cyanotoxins, PFAS, neonicotinoids, insecticides, VOCs, endocrine disrupting candidates, the personal care products, and legacy pollutants such as dioxin. We've now have many of these analyses capable of doing them in water, aerosols, dust, sediment, soil, tissue, human, animal, and plant, cells, and bodily fluids such as urine and spit. And some of them are metabolites, for example, like VOCs. Another area that we've gone into is we've started looking at imaging. So the right-hand image, and I forgot to get this on, is actually a published image. And this image here is what we have is this is a mussel. And this mussel, you can see that the left-hand side over here, the left-hand side has fluorescent dye. And these fluorescent dyes are actually where the plastics are, the microplastics are. And then we did MALDI imaging on that. And you see here, green and red, which we actually picked up.
Hey, Judy, you're
It's going out again. MDZ values and those plastics, thank you again. Those plastics, we can't say they're plastics because that, but we know they're associated. Whatever those masses are, they're associated with the presence of plastic in these. So we're looking at imaging, and we've also just recently got a paper accepted to look at imaging of microcystin LR in mouse liver. And finally, we are starting to look at imaging plastics in another way. And that's actually, and I spelled that, I'm sorry, Samir, I spelled your name wrong, but we're using XPS and Raman identification and imaging to do that. And to give you an indication, is XPS down here, is we can tell the difference in the types of carbon bonds, which will allow us to tell the difference in the plastic. And also potentially allow us to predict how things degrade. So that's one of the places we are going in the future. And this would be an imaging process. And from that, I will stop and take questions.
Thank you so much, Judy. So the last project, for those who don't, first, I guess I'll just quickly introduce myself. I'm Jamie Steve Thorsby. I helped organize the Water at Wayne seminars, and I am I'm going to be running the Q&A, but I'm a program coordinator with Healthy Urban Waters. But yeah, so if anyone has any questions, please feel free to send them through the chat. I really enjoyed your presentation. I have been working with Judy on the last project that she presented, and I've seen the results, but it's still really cool to just see them again. So I feel like I'm always learning something new, no matter how much I've seen all the results and whatnot. So, but yeah, if anyone has any questions, you can feel free to send those through the chat, or if you'd like to unmute yourself, you're also welcome to do that too. Yeah. Okay, it doesn't look like we have any questions. I guess I have one question, which was, what are some of your biggest challenges when you're, I guess, dealing with a new project, whether it be, it could just be technical challenges, it could be just, completely, coming up with new methods. I guess.
Yeah, I'd say the biggest challenge we have is different matrices. So today we might end up with urine, tomorrow fecal matter, the next day, it could be, then today we get tissue and it was basically put into liquid nitrogen, and that's great. And we can just slice or dice it or extract it. And then tomorrow we get tissue and it's matrix embedded. And we're like, okay, what is the matrix? So sampling, the earlier we get in on sampling, usually the more successful the projects are, because we can help people design that so that we understand what we're getting for matrix effects, and you can minimize them if we get in early enough.
Okay. Do you find with that, like if you work on projects, I know that you were saying that like you had three different kind of projects that you work on, whether it be like directly collaborating versus, someone just requests you to, analyze something.
Right.
I'm assuming, I guess I don't know this for sure, but I'm assuming the ones where you're directly collaborating is a little easier and maybe more interesting than, just getting it.
Well, it depends. It
depends.
It tends to be easier, but also once you have a method up and going and they say, okay, we're going to drop off urine and we want you to measure this. It's like, okay, we can do that. It's also easier from the standpoint if it's already been published too. When we write up like the 2 sentence for methods, we just refer to the other method. But no, the most important thing I was going to argue from an analytical perspective is that sampling. I'd say not the most important, but it's the first step, and it does really matter.
Mhm.
And people tend to people doing the bigger picture tend to forget that what you get out of it is going to depend on how you set that up. So understanding that and mapping that is really critical. We're looking at getting like 300 samples in from San Francisco. And it was like, I just ordered a new freezer and we're just gonna label it San Francisco freezer so they don't get lost, you know? So it's like,
Yeah.
those are things you think about after being like as old as I am. And you're sitting here going, okay, how do I do handle like that? Like these are gonna come in and they're gonna come in like every 4 months or 3 months. So
Mhm.
it'll be their freezer. So so we were just talking about, those are the things, you know. I'm most excited about imaging. If we could start imaging plastics and identifying plastics and where they are. The moldy has 100 micron spot, so that's a small spot, but it's not as small as one might want it to be. Whereas the XPS, we believe we're going to get 10 microns. So and be able to identify the plastics from either the Raman spectra from that or the XPS or both. And then that's the first step, is just identification. And then the second step would be is, you know, can we actually determine anything regarding degradation? And we think we don't see anybody using that. We see very few, not nobody, very few people using that instrument for that. And Samira's doing an excellent job bringing that forward. And so, yeah, he surprised me. He's like, look, I'm doing this. It's like, good. That's what I wanted you to do. But yeah, he's doing an excellent job. And like I said, hopefully we're looking at imaging the next 2 weeks tissue with high metal concentration to see if where the metals stay like in the tissue, if it's in specific areas.
Okay, very cool. We actually did get one question.
Okay.
And this is from an undergraduate research student who is currently working on a methodology for PFAS detection in soil and water using a bioindicator species. So they're wondering, do you have any advice for how I might direct my research in a way that best contributes to current work on PFAS detection, or any general advice on how you would stress test a new methodology?
Okay, so first of all, he said a bio, right? So I'm assuming
Bioindicator.
a bioindicator. So your first thing is you should actually really know what concentration your PFAS is at. So I'm going to say that you should be dropping off samples pre and post your experiments to our lab to let us measure it for you, so that you really know what concentration. PFAS is funny because it sticks to glass, it sticks to all sorts of things. So you're going to have to deal with that aspect of it. Just because you think you got it into the jar, oftentimes you don't get it into the jar. It amazes me that people, that's an experience you have because you're as old as I am, but things stick to the side, they stick all the way through. Every time you dilute them, if you didn't mix it right, if you didn't actually put it into methanol to start with and then move it forward for the dilutions, there's all sorts of tricks to working with something like PFAS, which is a surfactant. So if you really need to know where you're at. And one of the ways to really know where you're at is to have somebody else tell you that, yes, you are where you think you are. And if you're not, then you need to troubleshoot that. And so that's my recommendation, is that, again, going back to that simple, you really know the concentration in which your species is responding to. And that's going to be really critical.
Okay, very cool. Okay, that's very good to know. Did that answer your question, Caleb? Do you have a follow-up? Okay, I'm going to assume that that's all good. Okay, I'm gonna thank you so much for speaking, Judy. That was a great presentation, and you did a really great job answering all the questions that I had and that were submitted. I just want to really briefly mention that we have a Water at Wayne in 2 weeks, another one. It is on November 11th, and Dr. Peter Larson from the University of Michigan will be speaking on flooding in Detroit. the what, where's, and who's, and how water threatens public health in the D. So I believe everyone who has registered for the seminar, you should be automatically registered for the next one. But if you'd like, I will be sending out a link to this recording and more information on how to register for the next seminar if you didn't get it or if you'd like to forward it to anyone else. So, but yeah, otherwise, thank you everyone for attending, and enjoy the rest of your week and your weekend. Thank you so much again, Judy.
Thank you. Bye-bye.